Hi, I'm Bruce Bean. I'm a research neurophysiologist at Harvard Medical School and a co-inventor of the HotShot treatment for muscle cramping. I'm here to talk about what we know about how muscle cramps originate and how to treat them. You might think that the physiology of something as common as muscle cramping must be well understood. And you might think from seeing athletes on TV eating bananas or being given sports drinks by trainers that these things are well based in science. I certainly assumed they were before I got interested in cramps and started reading the scientific literature. But as I'll tell you, almost all of what we've all heard about muscle cramping turns out to be wrong.
My own interest in muscle cramps started when I was sea kayaking with my friend Rod McKinnon, and we were off the coast of Cape Cod on a cold fall day in heavy seas and a long way from any good landing spot. And both of us simultaneously started getting cramping in our arms and hands so badly that it made it hard to hold on to our paddles. That was a frightening experience, and it led us to start reading about the science of muscle cramping. We are both neurophysiologists who've spent a lot of time at conferences on muscle physiology, but we realized that we had no idea what causes muscle cramping. And so we did a lot of reading in the science literature over the next couple of years. And we discovered that most of the commonly accepted ideas about cramping, including those we had heard in graduate school and medical school, are now known to be wrong. So I'm going to tell you about what we learned and, most importantly, how it led us to a new treatment for cramping by doing experiments on ourselves. So, an overview of what I'm going to tell you that we learned is that muscle cramping is not caused by dehydration. It's not caused by lactic acid buildup in the muscles, and it's not caused by electrolyte imbalance in the blood. What we now know is that cramping likely originates from hyperexcitability of the motor neurons that control the muscle. So it doesn't occur from the muscle itself, but it's produced by the neurons that command the muscle to contract. So these are neurons located in the spinal cord, and specifically a population of neurons called alpha motor neurons, which send the electrical signals for muscles to contract. And we now know that cramping almost certainly reflects hyperexcitability of these neurons, these motor neurons, firing at much faster rates than they normally would. The excitability of motor neurons is regulated by very complex mechanisms, including feedback from the muscles, local spinal cord circuits, and I think especially critically, modulatory inputs from the brain that release neurotransmitters like serotonin, norepinephrine, and dopamine, and other agents that control the excitability of the motor neurons. What we ended up discovering was that cramping can be inhibited by stimulating a population of sensory neurons in the mouth and probably the esophagus and stomach as well, which can be done quite effectively using agents derived from a bunch of natural products. We call this trip channel stimulation. And this works through a pathway that ultimately reduces the hyperexcitability of the motor neurons that are causing the cramping to begin with. So, dehydration. We've all heard that the cause of cramping is being dehydrated, and that if you drink enough fluids, you can prevent cramping or stop cramping. Turns out this is wrong. And the work showing that it's wrong has been largely done by an exercise physiologist named Martin Schwellnus in South Africa, who's done a variety of experiments showing that neither dehydration nor loss of blood electrolytes cause cramping. The experiments shown here is from an experiment he did looking at triathletes during an Ironman competition in which he compared the level of dehydration and electrolytes in the group of athletes who suffered cramping during the competition with those who didn't suffer cramping. And what he found was that there was absolutely no difference in either of these things. So if he looked at the level of dehydration of the athletes who suffered cramping, they lost some water during the competition, but they lost no more water. They were no more dehydrated than the athletes who didn't suffer cramping. And for both sets of athletes, there was no change at all in the blood concentration of sodium. And in fact, the blood concentrations of sodium and potassium are clamped really well by the kidney. And so it takes extraordinarily extreme conditions for these to change. And it's not something that's going to change, even during something like an Ironman competition. So, from this work and a variety of other work that Schwellnus and other people have done, it's become very clear that exercise induced cramping is not, in general, caused by dehydration or changes in blood electrolytes. And in fact, this is something that if you talk to athletes who suffer from cramping, they know this very well because these are people, particularly elite athletes, who are very careful to maintain their blood electrolytes, to replenish their electrolytes, to maintain their level of hydration, and who nevertheless suffer from cramping. So, what does cause cramping? Well, the truth is we don't exactly know. And part of the reason is that there's no good model, neurophysiological model in an animal of muscle cramping. However, we do have some models of a very closely related phenomenon, which is muscle spasms. And so, for example, in people or animals with spinal cord injury, muscles will go into spasms, which is very much like a cramp. And in analyzing how spasms are generated, what neurophysiologists found was that the spasm comes not from the muscle, but from hyperexcitability of the motor neuron, from the motor neuron firing repetitive, what are called action potentials, which are generated in the nerve and travel down to the muscle to tell it to contract. These action potentials in the motor neuron fire at an unusually high rate and in an unusually maintained way during a muscle spasm. Important feature about muscle cramping brought about by exercise is that it almost always occurs in muscles that are being used repetitively and contracting strongly. It turns out there's a positive feedback loop whereby neurons come back from the muscle that's contracting back into the spinal cord to signal that the muscle is contracting. And these go through a positive feedback loop that actually causes increased firing of the neuron that makes the muscle to contract to begin with. Now, normally, this positive feedback loop is kept in check by various negative feedback loops within the spinal cord. But it seems like when there's extreme use of the muscle, that this positive feedback loop goes out of control. These negative feedback loops no longer control it. And the result is that stretching of the muscle causes more firing of the alpha motor neuron that makes the muscle contract. And that results in a very long lasting firing of the motor neuron, which results in a spasm or a cramp. These observations of muscle spasms in animals that result from uncontrolled firing of motor neurons fit very well with a parallel idea proposed by neurologists that cramping in people also reflects out of control firing of motor neurons, almost a kind of epileptic activity, but just in a small population of motor neurons in the spinal cord. And it seems that this uncontrolled firing of motor neurons can somehow be triggered by pushing muscles to their limits by repetitive use in intense exercise. A very cool thing about studying muscle cramping is that although it's hard to study in animals, it's actually quite easy to study in yourself. These are experiments that Rod and I started doing on ourselves. By stimulating a nerve going to a muscle repetitively, you can actually induce cramping in that muscle. And it's quite interesting. And this is something that athletes know, which is that muscle cramping is associated with heavy and repetitive use of the muscle. So, when you cramp, it's the muscles you're using heavily that cramp. It's not all muscles, which again is evidenced against the idea that cramping reflects dehydration or changes in blood electrolytes, which would affect all muscles. But that's not what happens. It's just the muscles being repetitively used. So you can make use of that principle to induce cramping in yourself. We use contraction of the muscle controlling the big toe. And so what you can do is you can set up a stimulating electrode over the nerve that goes to the muscle. And then every time you stimulate that nerve, you cause a contraction in the muscle. And you can record that contraction by electrodes right over the muscle. And that's what's shown in these records here. So, for example, here's a recording of the muscle contraction when you stimulate the nerve going to the muscle at different frequencies. So here we stimulate the nerve at 8 times a second for 5 seconds. And every time the nerve is stimulated, the muscle contracts and then relaxes most of the way. So you do that 40 times, 8 times a second over 5 seconds. And once you stop stimulating, the muscle relaxes completely. But now look what happens if you stimulate the nerve going to the muscle just a little bit faster, at 10 times a second for 5 seconds. Then what happens is that when you stop the stimulation, the muscle actually stays in contraction. It's gone into a cramp, and you can record that cramp by recording the muscle contraction, which in this case lasted for about 37 seconds. And here, if you stimulate the nerve going to the muscle at 12 times a second to stimulate it a little bit more, now the muscle goes into a cramp that lasts even longer for over a minute. So this turns out to be a really wonderful way to study muscle cramping because you can quantify the muscle cramping very nicely by the duration and the magnitude of the muscle of the muscle activity that you record here. And so this turned out to be a really great way of studying cramping and to start experimenting on ourselves for ways to inhibit that cramping. Rod and I started off by inducing cramping in our calf muscles, which worked very well. But if you've ever had an uncontrolled cramp in your calf muscle, you know that it can be extremely painful. And my wife and daughters were completely unwilling to become experimental subjects using this procedure. So, by reading the scientific literature, we realized that we could do studies just as well by inducing cramping of the big toe muscle. And this was a protocol that my wife and daughters and Rod's brothers and sisters were willing to join us in testing. And we spent a long weekend, one July 4th, testing all the members of our families using this procedure. And one thing we discovered was that people who tended to get cramping in their normal life, like my daughter Carrie, who gets cramping at her foot often when she swims, also tended to be much more easily induced cramping with the electrical stimulation and tended to have much longer lasting cramps, which was very helpful for then testing various treatments. As we were reading about the science of cramping, we also started talking to a lot of athletes who had experience with muscle cramping. Rod's brother Don McKinnon is a very serious amateur triathlete, and my sister Margot is also a triathlete. They told us about the folk remedy that they'd heard about from long distance cyclists and other endurance athletes, that drinking pickle brine could sometimes help reduce cramping. The idea apparently was that the fluid and salts in pickle brine were somehow just right for electrolyte balance. We got especially interested in this when we saw a paper by an exercise physiologist named Kevin Miller, who did an experimental study showing that drinking pickle brine actually had an effect on cramping. He did experiments very much like the ones we were doing using electrically induced cramping, and found that if people drank pickle brine just after being stimulated, then the duration of the cramp was considerably reduced from about 150 seconds down to about 85 seconds. And even more interesting was that this effect occurred within 30 seconds, which is far too short for the pickle brine to be effective affecting the electrolytes in the blood. So he didn't exactly know how it worked, but he speculated that somehow it might be working through some sort of nerve reflex. Thinking about how pickle brine might work led us to a hypothesis. The main ingredient in pickle brine is vinegar, which is acetic acid. And acetic acid is interesting because, as a so-called weak acid, recent work has shown that it's effective in activating particular receptors or ion channels in sensory neurons, and specifically two kinds of receptors called TRPV1 and TRPA1 channels. There are neurons with TRPV1 and TRPA1 channels in the mouth, and the esophagus, and the stomach that would be activated if you drank acetic acid. And then we realized that there are lots of natural plant products that are actually much more effective than the acetic acid and pickle brine in activating these channels. And so we decided to start investigating whether we could make combinations of some of these agents to get more powerful and longer lasting activation of these TRP channels, and then see if this would be more effective in inhibiting cramping than pickle brine. This worked remarkably well. We made a beverage from a combination of plant extracts containing the most powerful TRPV1 and TRPA1 activators that we could think of. And we found that drinking a shot glass full of this beverage was very effective at inhibiting cramping. So you can see here in this example, before drinking the beverage, we can stimulate the nerve and evoke cramping in the muscle very effectively, reproducibly. And then 12 minutes after drinking a shot glass full of this TRPV1 and TRPA1 stimulating beverage, which we called at the time flex beverage or TRP stimulating beverage, it was completely impossible to evoke cramping, even with higher frequency stimulation of the nerve. And what was even more remarkable to us as we did these experiments was that we found that often this effect of inhibiting cramping was maintained for several hours. And so, for example, in this case, even 4 hours after drinking a shot glass full of the flex beverage, it was completely impossible to evoke cramping by stimulating the muscle in the usual way. We studied this with toe muscle cramping, but then thought, well, we should really test this on the cramping of larger muscles. And so this is an example where we tested it on cramping of calf muscle, an experiment done on Rod, in which we stimulated his calf muscle at a high frequency. It went into cramp. This could be quite reproducible. And then in this case, 4 minutes after drinking the beverage containing the TRP stimulation, there was no cramping. And that was maintained for quite a while. So, how does this work? Well, the truth is that we don't exactly know. We're quite sure that the first step is the activation of sensory neurons in the mouth and probably the esophagus and the stomach as well by the powerful TRP stimulators in the beverage. From these sensory neurons, there are a number of nerves, including the vagus nerve, that carry signals up into the brainstem to a region called the nucleus tractus solitarius. And from there, there are nerves that go up into the rest of the brain to influence other regions of the brain, including regions that release neurotransmitters like serotonin, dopamine, and norepinephrine. We think these regions get stimulated by this pathway of sensory neurons. And we know that these regions have nerves that go back down into the spinal cord and release into the spinal cord circuitry transmitters like serotonin, dopamine, norepinephrine, and others. And we know that these transmitters can modify the excitability of alpha motor neurons and can help control whether they're going to fire excessively to produce muscle spasms or cramps. So we think that the level of these transmitters in the spinal cord is altered in such a way as to convert the uncontrolled firing that the motor neurons undergo to produce cramping back into controlled firing that produces then normal, well-controlled contractions of the muscle fiber. Well, we were very excited about how well this worked, and after coming up with this prototype beverage by experimenting on ourselves and our families, we thought it would be great to make a version that could be made on a larger scale for other people who might find it useful. So we began talking to people with expertise in making consumer products to see how we could do this. It turned out to be a lot more difficult than we expected because although our prototype beverage worked very well, it had a serious problem with the way it tasted. I remember very well a potential investor with expertise in consumer products who took a shot of our prototype and said, "My God, that's awful. That's like drinking hot lava." Fortunately, we were able to put together a very talented team of chemists and taste experts and formulation experts who were able to develop a combination of natural products that had strong trip channel stimulators that worked just as effectively at inhibiting cramping, but actually tasted good, unlike pickle brine or our various prototypes. That is HotShot. And with this quite good tasting formulation, we were able to do wider testing. And Daniel Craighead and Larry Kenny and their colleagues at Penn State did a controlled study finding that this formulation was very effective at inhibiting exercise-induced cramping, in addition to the electrically induced cramping that we'd used in developing it. This and other lab and field studies have documented the efficacy of HotShot for inhibiting cramping, and we've gotten really great feedback from athletes, including many people who've told us that they have less muscle soreness after intense workouts when they use HotShot before or during the workouts. Interestingly, after it became available for a lot of teams, we heard from trainers and athletes that a lot of athletes who never suffer from cramping uh, like to take HotShot also during workouts and competition just because of the way it makes them feel, which for me I describe as kind of a warm glow. So, to summarize, exercise physiologists have discovered fairly recently that muscle cramping is not caused by dehydration or a loss of electrolytes from the blood. And this is not to say that you shouldn't be careful to stay hydrated and replace electrolytes that you lose by sweating. You should absolutely do both those things for a variety of other reasons, but doing them is not going to prevent cramping. Cramping originates from the hyperexcitability of the alpha motor neurons that control muscle contraction. And the excitability of these motor neurons is controlled by spinal cord circuits that we don't completely understand. But what we've learned is that cramping can be very effectively inhibited by chemically stimulating sensory neurons in the mouth and probably the esophagus and stomach as well, which we can do very effectively by activating trip channels in those sensory neurons using agents derived from natural plant extracts that work through a pathway that ultimately reduces the hyperexcitability of the neurons that produce the cramping in the first place. So, after all the work Rod and I did together, experimenting on ourselves and on our families, it's been really gratifying to be able to work together with a really wonderful and talented team of people to develop a product that tastes good. And it's been even more gratifying to hear from so many athletes that this has really made a difference to them. What a lot of athletes have told us is that just being free from the fear of cramping enables them to go all out in training and competition in a way that they couldn't before. So I hope this can be useful to you or someone you know, and thanks for listening.